And now let's move on EEG, biomarker epilepsy and drug screening. And I'm very pleased to welcome mm, Alberto, who will give us an overview on his work. The floor is yours.
Okay, hi everyone. Thank you very much for involving me in this very important initiative. I'm not going to give a lecture on epilepsy and Q. I will simply share some information regarding our clinical and research activities, and I will also discuss a framework which I think is useful to think about how we can try to improve the care of people with Q, including epilepsy outcomes. So I'd like to start by saying that Q has a sort of an older sister called Angelman syndrome, who has been clinically described well before Q, and also has been the object of much more research, to the point that currently Angelman syndrome can be considered a model of the path toward precision medicine for rare neurological disorders, a path that is made by different steps or components interconnected with each other, which ideally could lead to more specific and better treatments. And so for Angelman syndrome, we have natural history studies, clinical management guidelines, we have a consortium for the validation of biomarker and outcome measures, we have a variety of different preclinical models, and we have, importantly, clinical studies evaluating the safety and efficacy of different treatments, including treatments specifically targeting the genetic mechanism, the neurobiological abnormalities underlying the disorder. Q syndrome currently lags behind in this ideal path toward precision medicine, although there are also positive news, as, for example, a specific treatment based on antisense oligonucleotides is in preclinical developmental phase. Why is that relevant for epilepsy and Q? Because, unfortunately, as we can see from our experience, epilepsy is very frequent, at least in children with IDIC-15. Probably in our cohort is too frequent, okay? But it remains often active despite adequate treatments, which often involve multiple pharmacological and non-pharmacological intervention. And this brings us to current treatment option, what currently can do. I will focus only on anti-seizure medication, the more traditional approach. So as you can see from this figure, there are actually quite a lot of drugs available for the symptomatic treatment of epilepsy. However, we don't know at present if there are specific drugs or mechanism of action that are more effective than others for Q. And in order to start to have a clearer picture of this, it would be really important to start to share information among different centers to get a sort of an overview of current practices and also to design proper clinical studies. And since we are here today to share thoughts about how we can improve things, including epilepsy treatments, I listed here 3 possible strategies, from the most simple to the most complex being, in theory, the most effective, involving treatment specifically targeting the genetic abnormalities. In our case, for example, treatments that are capable to modulate the overexpression of UB3A and other contiguous genes. This option is the future, hopefully a very near future, but there are other things that we can do in the meantime to try to improve epilepsy outcomes. The first is to look at novel anti-seizure medication, so medication that have not been developed specifically for Q, but have novel and different mechanism of action that can benefit at least some children with epilepsy. And the second is drug repurposing. So basically, we should look at drugs that have already been approved for other clinical indications and see whether they might exert specific effects for Q. So regarding novel anti-seizure medication, I will mention CBD as an example, or cannabidiol. Its use has been reported in a small group of children with Q, with some of them showing a significant reduction of seizure at the end of the treatment period. Along with other Italian colleagues, we are about to report on the effects of CBD in another group of people with Q with slightly better results. But the main point is that if you could have more drugs with different mechanism of action that can benefit at least some children who have not responded to previous treatment, then we'd be ideally be able to reduce to some extent the burden of epilepsy. particularly if we will be able also to predict treatment response to some extent, to personalize treatment. Regarding drug repurposing, it is important to cite the work of Professor Later and colleagues in the US who developed a Drosophila model of Dovzhenko epilepsy. Drosophila is the fruit fly. They screen a lot of different drugs in this model, showing that there are at least 3 different mechanisms of action and 3 different classes of drugs for which safety profile has already been established, who in this model are able to suppress seizure activity and to do so more effectively than other traditional anti-seizure medication, which is interesting. Drosophila is, of course, a very simple model with all sorts of limitations, but it's interesting to know that, for example, drugs that are currently used for depression might exert specific anti-epileptic effects for Dovzhenko. And this is something that might be tested in the context of clinical trials. It is already clear that in order to move things forward, we need joint efforts at any level. I would like to add a couple of thoughts about that. The first is that our goal is personalized medicine. Then we will likely need novel objective biomarkers, ideally non-invasive and easy replicable. And EEG is potentially a useful source of this kind of biomarkers. We have an ongoing project where we are using high-density EEG to study different forms of epilepsy, including epilepsy associated with Angelman syndrome and Dovzhenko syndrome. And we are looking for syndrome-specific profiles of brain activity, in particular function and connectivity, so the way different brain areas communicate with each other, and also markers of epilepsy status. So basically, we are comparing children with the same genetic condition with and without epilepsy. And hopefully, these could also serve as novel markers for evaluating clinical evolution and treatment response. And second, I think that would be really important to set up a network of different preclinical models, because each model has its strength and limitation. We'll need to try to get the best from each one. So, for example, Drosophila is very well suited to screen drugs, because we can screen hundreds of drugs, okay, in this model. And we can use Drosophila as a sort of filter. And what comes out from this filter could be tested and replicated in a more complex model, ideally a mouse model of Dovzhenko epilepsy, and also patient-derived neural networks, a model that Dr. Sapulano will discuss in a minute. So to conclude, maybe we should think about this agenda or path toward precision medicine in a more circular rather than linear way. So we need to start from the children, the families, and set up the agenda together from the start. Then we should look at all sorts of clinical and genetic data. We need to better characterize clinical manifestation and see which manifestation impact the most on quality of life. Then we can use genetic, but also EEG and MRI data to extract novel biomarkers. As I said, we need multiple different preclinical models, and hopefully this could accelerate drug screening and development. And in the end, we may be able to come back to the children and the families with something more valuable to offer. This is only a fraction of the people that make our work possible, and we'll be very happy to share this network with you. Thank you very much.
Thank you, Alberto. Very important points. Let's discuss it later. Now I will ask Monica to give us some of her to share with us some of her experiences of electronic clinical features in DAP50Q epilepsy.
So good morning, everybody. Thank you very much first for the organizers of this conference, and well, especially Verena and the whole association of the parents for organizing this excellent conference. I'm really very impressed. And thank you for the invitation to present our work, which dates a little bit back. And so I will present you this work we did in 2019 with one of our fellows in the department. So I'm pediatrician and clinical neurophysiologist, pediatric neurologist, specialized in epilepsy of the very young children, and I present you what we had observed. I have to say, in 2019, what was described in the literature was that epilepsy in DUP15 syndrome was in interstitial duplication described at a percentage of 25%, in isodicentric in 65%. Epilepsy was described as of early onset, and treatment was mostly refractory with a more severe course in the isodicentric one. Concerning EEG, there was already a notion that there was excessive beta activity during wake state in these patients. When in a study where they compared the background activity to autistic age and IQ matched children and to healthy, typically developing age matched children. So we asked what questions are eventual to answer. So we thought it could be interesting to see how do seizure types and epilepsy evolve with age, and how do EEG patterns change according to different vigilance states and to age. So we undertook this study, which was a monocentric retrospective study at our hospital in Paris, France, a tertiary referral center. Inclusion criteria were the diagnosis, cytogenetic diagnosis of duplication 15q syndrome, and the presence and the availability of EEG recordings. Of course, the population is biased. All these children are DUP 15 children with epilepsy, so there's not one without an epilepsy. So we had a look on clinical data, pregnancy, birth, development, behavior, autistic features, age at onset of seizures, seizure type and seizure semiology, the epilepsy syndrome when applicable, and response to anti-seizure medication. We evaluated the EEG activity, the background in wake and sleep, in the interictal and in the ictal state, and we undertook power spectrum analysis on 10 sensors on different locations, so 10 electrodes in frontal, central, temporal, occipital regions, both sides. So we included 12 patients fulfilling the inclusion criteria, 8 males and 4 females, and 5 patients presented with interstitial duplication, and 7 patients with isodicentric duplication. So here is a table on the clinical data. The follow-up was from age 4.5 months to 7 years, with a follow-up period median of 8 years. In all children, family history, pregnancy, delivery was uneventful. All had a psychomotor retardation and autistic features. Epilepsy onset was not different in the two groups. You see here the interstitial duplication and the isodicentric duplication. Onset of, in our group at least, was not different, with a median of 9 months, 8 months, which is much younger than in the studies we just had heard before. But this is, again, as we discussed, a problem of it's just a little population, a little cohort, and it would be very interesting and important to have that a little larger spectrum. So seizures recorded, not observed, recorded, were epileptic spasms in 7 of the patients, tonic seizures, myoclonic seizures, atypical absences, focal seizures, and generalized tonic-clonic seizures. The epilepsy syndrome, we had 4 children with, that was still called WES syndrome. It's now infantile epileptic spasm syndrome. And 4 Lennox-Gastaut syndrome, with 2 de novo and 2 children evolving from infantile epileptic spasm syndrome. The outcome was much better in the interstitial group, with 4 of the 5 children becoming seizure-free. Only 1 drug-resistant Lennox-Gastaut syndrome, which has at this time not yet described, but I've seen, I can't remember who, but there was one who described also in the interstitial group pharmacorefractory Lennox-Gastaut syndrome. And as it was also said several times, the outcome is less favorable in the isodiacentric group with mainly pharmacoresistance. Then, so here we see just an example of a recording of a peculiar seizure type. This you can see brief tonic contractions of the four limbs. They do not really fulfill the criteria of epileptic spasm, apart from the organization in clusters, and also the well, I stop here, gaining time. Apart from the cluster, and also the ictal EEG correlate as a spike, polyspike, posteriorly predominating a burst concomitant of the brief tonic contractions. So this is particular pattern, and that was observed in 4 of the 5 children presenting with infantile epileptic spasm syndrome, because apart from this, they also had classical epileptic spasms. And this pattern could be observed also in other children. So here are these 2 other examples, sometimes also without a motor correlate. And also interesting was that in the 6 patients diagnosed infantile epileptic spasm syndrome, hypsarrhythmia was either absent or atypical. As you can see here, why did we term that atypical? Because the amplitude increase was moderate, so we could still interpret the EEG at an usual sensitivity, and the slowing of the background was also moderate. Here you see a typical hypsarrhythmia. So no typical hypsarrhythmia in all of these children with infantile epileptic spasm syndrome. Then we had a look on the interictal EEG features, and here on the EEG you see Alberto had shown it also, but not really said. So you see a very peculiar aspect, this excessive beta activity in the awake state, which was present in 11 of the 12 of our children. And here you see in sleep it diminishes, and you can even see physiological sleep patterns, sleep spindles. Sorry, this is a presentation in a trending, 2.5 hours recording. This part is wakefulness, and this is in between asleep. And it's a trending in a kind of density spectral array, which represents graphically frequency power. And you can see here these bands, which represent an excessive activity around 70 hertz, which diminishes in sleep and comes back in wakefulness. And then we investigated this by a spectral analysis. And so here you can see that, and we had a look according to age. So 4 age groups, the under 1 year, 1 to 2 years, 2 to 6 years, and over 6 years old. And you can see here is in red, these are the frequencies, the power of the frequencies in red is during the awake state, and this is in sleep in the blue line. And what you can see in all age groups, that all frequency is higher than, here it's 17, and later on it's 26. There are much more, power is much more increased in the awake state, which is statistically significant. Then we had a look in the same groups on this frequency power on different electrodes. And what you can see in younger children, it is all the different colors. You can see there are the colors according to the sensors. Green, blue is in frontal, rather frontal and anterior elites, and violet and red, blue is in posterior areas, and in younger children, it's rather diffuse, and when you get older, it's clearly predominating over the frontal anterior regions. Then, in summary, this is Marie-Thérèse Dangle, who undertook this study, fellow at the time. We observed in our small 12 patients study group as main epilepsy syndromes, infantile epileptic spasm syndrome and Lennox-Gastaut syndrome, both occurring more frequently in the isodarcentric duplication. In interstitial duplication group, focal epilepsy was rather observed. And the seizure control that has also been described, and we heard it several times today, is less frequently observed in the isodarcentric group compared to the interstitial group. And in infantile epileptic spasms, hypsarrhythmia is always typical or absent. And the pattern, there was observed a particular pattern of posterior predominating spike and polyspike bursts, all observed in the isodarcentric patients. We confirmed the previously described increased beta band power during wakefulness, and we added a new observation that these fast activities decreased in non-REM sleep, and they shifted from diffuse to more anterior localization with aging. And I think this is a really, in a particular clinical context, it's a recognizable electroclinical pattern, which helps at least 6 years ago. It was very important in the guidance of identifying duplication 15q syndrome in epileptic children. We had a discussion during the break. Today it's perhaps less important because we rather quickly go to genomic evaluation, but there was a gap when we didn't do ARIES anymore, but not genome was too complicated, so there was a gap several years where it was difficult to find, or it took longer to find the underlying etiology. And then, last but not least, this increased beta power in the awake state is probably clearly a biomarker, and this can help to understand the underlying disease mechanisms and can potentially help monitor responses to emergent therapies. So thank you very much for your attention.
So, thank you very much, Monika. And now the panel is open for discussion and questions. Yeah.
So I've got a question about the treatment. Do you also treat just if there are just some special EEG pattern, or just if they have also clinical seizures? Is that a better outcome to treat early? And yeah.
Well, I will answer these questions globally as a pediatric epileptologist, but probably it's very specific in 15 duplication. But generally, we know what we do. We treat. The most important and to treat is the presence of an epileptic encephalopathy, because this is, we know the concept of this epileptic encephalopathy that contributes to degradation, behavioral problems, and many other problems by the fact that there are many seizures active and an encephalopathic pattern on EEG. So this really, particularly in Lennox-Gastaut syndrome and epileptic spasms, this has to be treated. It has been shown that has an impact on cognitive evolution and on development and on behavior, and also the treatment delay is very important as well. We know the longer the epileptic encephalopathy persists, the less better is the outcome in terms of cognition and response to treatment. If they're, for example, epileptic spasms, they last for a month. It's much more complicated to stop them.
So I wonder if it's good to recommend an EEG surveillance for these children, because some just start with the seizures clinically at the age of 12 or 15 years. And is it, do they benefit if we know the EEG pattern earlier, to start treatment earlier or not? Because normally you wouldn't recommend an EEG if there is no clinical seizure.
Well, I have not so much experience in older children, but I would think, I mean, I don't know the literature on that, if it's recommended, but I would think there's always something clinically. There is, from the clinical situation, you have criteria to ask for an EEG. I don't think a regular surveillance of EEG would show you what will happen in the future. It shows you what happens at this moment, and you could have 1 week later beginning of an epilepsy, and you wouldn't see it with a screening. But there are always clinical signs, I think so. They're either seizures that you can see, or regression, stagnation of development, or behavior problems increasing. But yes, but probably listen to the parents.
Yeah, the expression.
Yeah, I think you raise a very important point. I think that I have in mind a few children that we are following up, and they do some EEG from time to time. And for some of them, we are seeing that EEG abnormalities are kind of builds up over time. Okay, so you start with a almost normal EEG, and then you can see that EEG abnormalities start to be present in EEG and tend to worsen over time, until you finally have clinical seizures. The problem is that we don't have anything, we don't have evidence that there is something that is capable to prevent seizure. So basically we tend to treat seizure when they occur. Okay, but I think that we also discussed that at times seizure onset is quite late, happen quite later on. And we also have seen children with seizure onset at puberty, okay, without a previous history of seizure. So we can also think about these periods of seizure freedom as potentially windows for prevention, if only we could have something that is capable to do so. So it is a very, I think, important challenge. And there is, for example, the model of tuberous sclerosis complex, where they start thinking about, okay, we treat the patient since the first EEG abnormalities, because we can probably improve epilepsy outcomes later on. But I think the most important thing will be to find evidence that there is something really superior to something else for this kind of epilepsy.
Comment?
I think, as far as I know, from the G15Q Alliance in the States, they say we should do a 24-hour EEG at the beginning, to have at least a baseline to compare it then if it worsens. So that's the recommendation that they give from their professional advisory board. But I mean, it's maybe different in Europe.
Okay. If we do not have more questions, something in time. Irina, I want to ask you to come up. So I will thank all the participants, all the listeners, all who contributed to this symposium. I think it was a great symposium, the first DAP50Q symposium in Europe, and we have to continue this. I have 2 things to say. The first is, afterwards, please come here. Let's make a photo. Let's post it, and to show the world we are here. We are fighting for research and for the DAP50Q patients. The second thing I would propose is that all specialists who are interested in a concrete collaboration, that we come together after 10 minutes after the ending of this session to think concretely how we can go further. What are our projects? We have to think of a European project, right? Okay.
I'm a little bit overwhelmed, but I really thank you so much that you have all come. No, no. But for me, it's so important that you have all made the way. And you don't know me, I mean, they know me, but you have all taken your time out of your busy schedule. You have traveled here and you made that first symposium here, and I think that's so important, a message for our parents that there's something going on in Europe, and so thank you all very much.
Verena, you made it. You made it. Thank you for all what you have done.